Hello, applicants. We were asked to provide some information about what we do in our lab, so you have an idea in case you want to do a rotation or you want to come to work with us. I'm a professor at the Star Student Transplantation Institute. My name is Adrian Morelli, and I work in the area of transplantation immunology, more specifically, the role of extracellular vesicles in the biology of transplantation. And more recently, even in fetal maternal intolerance. So cells, this is one cell, another cell, communicate to each other, not only through cell-to-cell contact or release of soluble mediators, but also through the release of these tiny vesicles that they contain proteins, lipids, carbohydrates that are transferred from one cell to another. So more specifically, we've been working on this question that has been around in our field of transplantation for the past 70 years. That is, in what form the donor antigens get from the graft, from the transplant, to the place where the leukocytes are. So to initiate the process of a graft rejection. So the hypothesis, the current hypothesis, is that the cells from the graft, in this case the graft and all the donor cells are in red, once the graft is transplanted, the leukocytes that are in the graft migrate out of the graft. They go to the secondary lymphoid organs like lymph nodes of spleen of the recipient that are in blue here. And then they interact with the recipient's leukocytes, T cells or B cells. And this is the way by which the T cells and the B cells of the recipient recognize the allograft. And this is what starts graft rejection. But there's a problem. Even if we inhibit the migration of the donor leukocyte for the graft, we still can generate graft rejection. So in recent years, there is some work on the release of a cellular material from the graft in the form of small vesicles that transport the donor antigens from the graft to the lymph node or spleen of the recipient. So there are this field of transplant immunology started more or less 4 years ago. And there were these two papers, one from our group and the other from a group of our colleagues in Harvard. But they demonstrate that the graft release extracellular vesicles that deliver the alloantigens and a lot of inflammatory signals that trigger the rejection process. A little background of these extracellular vesicles. These are released by practically all kind of cells. There are some of these vesicles released from the cell by the cell membrane, they're called microvesicles. Others are released from these structures that is a compartment of the endocytic compartment of the cells, and these are very tiny, that are called exosomes. Particularly exosomes are very interesting for our work because they have on the surface all the donor antigens that elicit graft rejection. So it's a very good mechanism to deliver donor antigen to the recipient and to start the rejection process. And these vesicles, we know that interact through different mechanisms. Some of them remain attached on the surface of the acceptor cells. Others are internalized like a virus, and then release their intraluminal content into the cytosol of the target cell. And these vesicles not only have proteins, lipids, carbohydrates, but they also carry mRNA, microRNA, non-coding RNAs that can affect the behavior of the acceptor cell. So these are the typical experiments we do in our lab. We do heart transplants in mice. We mostly work in mouse models. In general, these heart transplants are done in the abdomen, not in the thorax, like in human beings. And we follow, obviously, the release of the donor leukocytes into the draining lymphoid organ. In this case, is the spleen. Here you have one of the donor cells in green. But we also follow the release of vesicles from the donor to the leukocytes that are located in this secondary lymphoid organs. Here you have an example. This is a recipient cell that after a heart transplant received a cluster of small vesicles that express the donor alloantigen that was labeled with gold. This technique is called immunoelectron So we know that this is a mechanism of transfer of these vesicles between the donor and the recipient. So we also do other type of model like a skin allograft in mice. And in this case, the skin allograft has donor leukocytes, they are yellow. And we can show you that at different times after transplantation, we cannot find the yellow cells as predicted by the hypothesis. But what we can find is that these tiny vesicles that were released by the skin allograft into the lymph nodes of the recipient. This is the donor vesicle expressing the donor MHC, again by immunoelectron microscopy. The different ways that we can produce these very tiny vesicles. One mechanism is to get an allograft and make the allograft to release the vesicles ex vivo. Another is we can produce it from culture supernanthers of cells that we keep in culture. And after several steps of ultracentrifugation and filtration, we come up with these big vesicles or small vesicles that we can label with different colors. And we can make movies in animals that are alive. That's called intravital multiphoton microscopy, by which we can follow these vesicles in vivo. So we can generate vesicles, in this case red. We inject the vesicles in the foot path of this mouse. And then after a few minutes, we can track the traffic of these vesicles to the closest lymph node, that in this case is a popliteal lymph node. When the animal is alive, when anesthetized, we open the area where the popliteal lymph nodes is, we expose the popliteal lymph nodes, and with a multiphoton microscope, we can make a movie. And this is what I'm going to show you here. And this is a case where we inject red exosomes in an animal where the cells in the lymph node were pre-labeled in green, doesn't matter what it is now. And you can see after a few minutes that the cells from the host animals are uptaking the red exosomes, the red vesicles. You can see that more detail, different type of approaches to make these movies. This is the surface of the lymph nodes that have captured these vesicles that were injected. The different population of macrophages, some of them internalize a lot. The medulla of the lymph node, others internalize less, but they transport the vesicles to the inner part of the lymph node. So we can do a little more complex experiments in which then we can transfer B cells or T cells and see how these B cells or T cells in different colors recognize the fully allogenic vesicles that we inject in the food bath of these animals, again by multiphoton microscopy. All this is done when the animal is still alive and exercise. It's a very cool techniques. And we can make things a little bit more complex and close to humans. We can generate humanized animals. This is a technique we use in our lab. So, and in these humanized animals that are mice that have chimeric human leukocyte, we can do human skin transplants in the back. And then we can follow the traveling of cells or vesicles that now are humans into a humanized lymph node and do similar experiments that I showed you before. So we have a lot of collaborators inside the Starson Institute, but outside, and we make our institution. And we are supported by grants from the NIH and fully supported also by our institute. That is one of the leading institutes in transplantation in the world. Well, thank you very much, and I wish you the best. And my doors are open always for the students if you have a question. Bye.